All right, this is going to be my second lecture for Monday. What we're going to do here is we're going to move on. We were talking about topoisomerases earlier, so we're talking about different things that happen with DNA. And what we're going to talk about now is DNA packing. So if you were to take the DNA in your cells and stretch it out, let's go ahead and make myself a little place to write. There we go. So if you were to stretch out the DNA from one of your cells, put it all end to end, it's actually 46 different pieces, right? But you ran it out end to end, it would be 2 meters long. From one of your cells. All right, and so that happens, it could be 2 meters long, but it has to be compacted down into a nucleus that's going to be a micrometer across or less. So micrometers could be 1 times 10. Here, I'll use the E thing. 1 times 10 to the negative 6 meters across. So your DNA has to be really compacted down a lot. Now it's thin, so it's long, but it's thin. So it does fit, but it's going to need to be stored in there. It's going to need to be compact on the size, and then it needs to be, of course, organized in such a way that genes can be expressed and it's accessible. So it can't be all in there in knots and things like that. Think about, when you have a bunch of yarn and it gets out of the ball form, that often it becomes a mess really quickly. So to accomplish this, there are several different layers or different levels of organization that occur. And the most basic form is that the DNA gets wrapped around histones. So the most basic form of packing is that your DNA gets wrapped around histones. Okay, and when we talk about histones, there are multiple histones. What we see right here, a histone core. Okay, and it's made up of, you can see that there should be 8 different histones in here. We've got pairs, 2H3, 2 H2Bs, 2 H2As, and 2 H4s. I've got all of them. Okay. So a histone core is made up of 4 types of histones. And there's 2 of each. What's interesting here, so my colleagues down at Virginia Tech I work on drugs binding to proteins essentially, but another main project in the group is that they're looking at mutations in histones that cause DNA to bind more or less tightly. And one of the things that they look at is this. You see how the H3 kind of sticks out here? It kind of has this long piece. And where we have one here and it's headed back. And that H3, that chain on the H3 histone has a bunch of positively charged amino acids. This is where I'd ask you what the positive amino acids are. And you would tell me that they were lysine, arginine, and histidine. And so these positively charged amino acids are either going to be arginines or lysines. Are not, you don't really find the histidines here so much. But those will wrap around the DNA. So the DNA will wrap around this kind of ball in the middle. And then this piece here can wrap in and kind of wrap over it and hold it in place, which is really interesting. Kind of fun stuff. All right, but then when DNA wraps around that histone core, it looks like this. So what we've got here is we've got DNA, it's going to wrap. What have we got? 1, and then 2. I think I've seen it said 2.5 times, but when I look at this, what do we got down here? Yeah, so we're not getting to see all of it here. So we go 1, and then 2. I've heard 2.5 times, and what that does is it shortens the length of the DNA 7 times. So DNA wraps around the histone core, and this shortens the length of the DNA by 7 times. Okay, and when you have the DNA wrapped around a histone core is called a nucleosome. Okay, so this is a nucleosome, and so it's the histones and the DNA is the nucleosome. So here we have a histone core and nucleosome, and then we have this DNA linker part. So from here to here would be a nucleosome. Let me draw that. So from here, say, to here is a nucleosome. And you have this linker bit, and what we're going to see is some electron micrographs. We can actually see them below. You have electron micrographs where you see, beads on a string is how they kind of describe this, where you've got the nucleosome cores and you've got linker DNA in between. But there are higher levels of organization, so you don't feel it's not really it's not in the cell like these spread out beads on a string. And what you end up with is more, here's what's called the 30 nanometer fiber. That might be a second level is a proposed second level of organization. And if you read the literature on this, which I've done to better understand what other members of the group I work with at BT are doing, this stuff is really complex. It's really kind of there's some kind of amazing, there's a lot of other proteins involved that hold different parts of the genome together, such that you will have pieces on a chromosome that are not close to each other in sequence space, but things loop around such and are held in place such that they end up being close kind of in a region of the cell that is, they're close in three-dimensional space, but not in sequence space. It's kind of like protein stuff. But there would be proteins that would hold that together. So I don't know how much this 30 nanometer fiber is out of a textbook that it might be 10 years old or something more. In terms of the stuff I see in the group, we don't really talk about that level of organization. But you think about, things where these histones will stack, or the nucleosomes will stack, and kind of think about how loosely they're kind of held together. And then if we look here, what we've got is this is kind of another proposed diagram of multiple layers of our levels of organization. So we've got our DNA, we wrap around histones to get the beads on a string. So we have our nucleosomes. Our nucleosomes end up in that 30 nanometer fiber. That fiber can then form the loops. These loops can form rosettes. The rosettes can form coils. Then you get a bunch of these coils together to make two chromatids. So one chromatid and another chromatid. And so this is a hypothesis of how these might go together. There may be some evidence for that. I'm not sure what that evidence is, but just want to give you a feel for that. What we are going to focus on now really though is just this beads on the string sort of nucleosome, and so you need to know about what a nucleosome is, know that there are 4 histones that go into it, and then we're also going to talk about, so we can end up with DNA, well, we can end up with mutations in amino acids that would cause like positively charged amino acids on the nucleosome to be mutated to something that wasn't positive. And we know that DNA is negative, so if we take away positives on the surface of the nucleosome, then the DNA is not going to be as attracted to it, and then it will be more accessible for transcription. Enzymes will be able to get at it if it isn't wrapped as tightly. Let me go ahead and write that down here. So DNA is held to the nucleosome, or to the histone core, let's say, partially by attraction to positively charged amino acids. Okay, these positively charged amino acids can be removed through mutation, and you can also remove them through post-translational modification. Okay, we'll talk about that in a second. We'll talk about specific ones in a second, but if you remove those, the DNA is not bound as tightly, and it will be transcribed more. So if you remove those positive charge or negative charges are added, the DNA will bind the histone core less tightly. And it will be more accessible for transcription. Where transcription is we're going to make RNA and then we can make proteins from that. So when we talk about mutation, mutations in histones, there are mutations to histone amino acids that are associated with cancer. So you have a histone that ends up being mutated so it doesn't have as many positive amino acids on it, so it has fewer lysines or arginines, and the result is that the DNA on the histone is not as tightly bound, and it's more likely to be transcribed, and you get things that are transcribed that cause the cells to continue through the cell cycle and keep dividing when they should stop dividing. Let's talk about some of these post-translational modifications, and what I'd like to do is let's focus on lysine because it's simplest in terms of chemistry. So if this was a lysine side chain on a histone, 1, 2, 3, 4, and then we have an NH3 right at pH 7, that's going to be an NH3 on that. Well, you could imagine that you could acetylate it. So we're going to add an acetyl group. Gosh, I don't even know where that acetyl group's coming from for sure, but I'm going to guess it's going to come from a CoA. It's a great thing that has an acetyl group on it. Okay, so if there are enzymes that actually go in and specifically change these amino acids, so that you'll have that happen in certain regions. You'll have the amino acids on the histones change, so those particular regions, those particular histones, the DNA that's bound to them will actually be transcribed more. There are genes that need to be transcribed more than other genes. And so these are one of the things. This is one of the things that happens. It's a normal, healthy part of what a cell does. So these things can become acetylated. Hopefully you remember, if we go ahead and we add an acetyl group to a nitrogen, we're going to get an amide. And remember that amides don't have any charges on them. Okay, so what we've done here is we've removed that positive charge, and this one right here is going to bind to DNA more weakly. Okay, and this binds DNA more tightly. And this one on the left binds DNA more tightly because it has a positive charge that's attracted to the negative charge of the DNA backbone. You can also have methylation events that occur, and that also seems to affect whether things are positively charged or not. So we can end up with methylation. This is a pretty obvious example. Methylation is not as obvious because if I put some methyl groups on here, I mean, it is something that happens. You end up with trimethyl here, and we put trimethyl on it. It seems to me that it would still be positively charged, but I guess the methyl groups get in the way. We're not going to worry so much about that. But I mean, you end up with trimethyl lysine. But let's think about lysine and what happens if we phosphorylate a lysine. Okay, so you could put a phosphate on this. Well, let's not even do it to a lysine. Let's do it to a serine. Imagine you have a serine, right? Serine and tyrosines are things that are often phosphorylated. So we've got a serine, and we can have ATP here, and we can make this. And now we've got negative charges sitting on our histone, and those are going to push the DNA away. So the DNA is going to bind this at the right more weakly again. And then we'd have ADP over here. So once again, binds more weakly, binds more tightly. So I would expect that you would know these as examples. Maybe on the study guide it asks you to know an example of one of these. I was to give you something that was obvious in terms of charges. I'd like you to be able to reason through binds more weakly, binds more tightly, based on the fact that you know DNA has a negatively charged backbone, and that it's going to be more attracted to positive charges or neutral charges, positive charges over neutral charges, and certainly neutral charges over negative charges. Neutral charges is no charges. All right, well, I'm going to stop there. That's what I wanted to talk about for histones and DNA packing. I may add a little bit more. I'm here with grad students that are working on at the conference, with some grad students from the group that are working on that project. If they have a cool video or something, I might share that a little bit more in the next lecture. Hope you guys are having a great day, and I will see you later.